Aspergillus is a genus of fungi that you encounter more than you may realize. Along with being important to the production of products you might be familiar with, such as soy sauce and sake, it is also estimated that we inhale hundreds of airborne spores called conidia daily. Imagine blowing on a dandelion. The feather-like seeds that fly off and disperse into the air are similar to the conidia. The microscopic conidia easily detach from the fungi and float around in the air, which makes them easy to inhale in our daily life. Aspergillus fumigatus is a species of particular interest since it's commonly found in the environment, such as in soil and air. Despite its many useful applications, this species is also considered to be an opportunistic pathogen and can pose a huge threat. Most people have robust enough immune systems to combat the conidia we inhale daily. However, that is not the case for people with compromised immune systems. Aspergillus infections, generally referred to as aspergillosis, mostly affects the lungs, as airways are the main point of entry for conidia. These infections may cause fatigue, weight loss, coughing, sometimes coughing up blood, and ultimately death if not treated quickly. But what makes Aspergillus so dangerous? A major virulence factor, a quality that makes Aspergillus so potentially harmful, is that it grows best at 37 degrees Celsius, the same temperature as the human body. So, if Aspergillus has the chance to colonize anywhere within your body, the most common area being the lungs, it will grow rapidly, which is why it's necessary to treat aspergillosis immediately. Therefore, the thermotolerance of Aspergillus, that is, its ability to survive at temperatures of 37 degrees Celsius and above, is a notably interesting and important virulence factor. The aim of my project is to identify genes associated with thermotolerance, or the ability of Aspergillus to grow at different temperatures, in order to elucidate the role these genes play in the virulence of the disease, which may ultimately help to better combat aspergillosis. I will perform a genome-wide association study in order to identify these genes. This involves testing over 100 different Aspergillus fumigatus mutant isolates. The mutant isolates differ from each other by a small variation in their DNA sequence, but this small mutation can drastically affect certain characteristics. Aspergillus grows best at 37 degrees Celsius, and as the temperature increases, the growth becomes increasingly more limited. If an isolate has a mutation in a gene responsible for thermotolerance, the ability of that isolate to grow at certain temperatures will be affected. I have identified a threshold temperature of 55 degrees Celsius, at which most Aspergillus will be unable to grow. However, out of the 100 mutant isolates I am testing, I am expecting to identify a few that will have the ability to grow at this high temperature. Since normal Aspergillus are unable to grow at this temperature, the ability of these isolates to grow at the high temperature must be due to a mutation in a gene allowing for this atypical characteristic. The mutation must occur in a gene responsible for thermotolerance. By comparing the DNA sequence of these isolates to a typical Aspergillus DNA sequence, we can identify where the sequences differ or where the mutation occurs, and thus identify a gene responsible for thermotolerance. Each isolate will be grown in a petri dish containing potato dextrose agar media, which provides all of their nutrients essential for survival, so the only limiting factor affecting their growth is temperature. There will be 4 replicates for each isolate. They are sealed with aluminum foil to keep light out, since UV radiation interferes with their growth, and incubated at 55 degrees Celsius for 1 week. After the incubation period, the area of growth for each isolate replicate will be found using a scan colony counter from Thermo Fisher. The areas will be averaged, and the isolates with the highest and lowest growth will be sent out to have their genomes sequenced. These sequences will be compared with each other, as well as the complete genome of Aspergillus fumigatus on NCBI, a public genome database. Differences in sequences will be identified, and these discrepancies that occur are expected to be located in genes responsible for the thermotolerance of the fungi. At this point, we will have identified one or multiple genes of interest. To further support our findings that the identified gene is involved in thermotolerance, our lab collaborators at the University of Tennessee will knock out this gene using CRISPR gene editing. This will create a set of isolates identical to the ones we have sequenced, except this time they will lack the gene that we suspect allows them to grow at the high temperature. We will again grow these isolates at 55 degrees Celsius, and if this gene is involved in the thermotolerance, it is expected that after it is knocked out, the isolate will no longer be able to grow at high temperatures, since it will lack the functioning mechanism to do so. And then we can confirm the gene of interest is in fact involved in Aspergillus fumigatus thermotolerance.